All right, guys, here we are in section 8.3, which is DNA replication. Our key concept is that DNA replication copies the genetic information of a cell. Our vocabulary, we've got replication, complementary, replication forks, DNA polymerase, and semi-conservative. Our essential questions: what is a process by which DNA copies itself? How does DNA replication work? And why would maintaining the exact genetic code in all cells be important? Again, please make sure at the end of this that you go back and you're able to answer these essential questions. So copying the code. DNA copies itself through a process called replication. Now, if you remember correctly, we have our DNA strands. We have two strands in DNA. Remember, it's a double helix. It's one set of nucleotides going one way, one set of nucleotides going the other way, and then in the middle, we have those nitrogen bases that form the rungs or the steps of our DNA molecule. Our DNA is a double helix. So the information of each strand in our double helix has all the necessary information to reconstruct the other half because of these base pairing rules. If you remember correctly, A always goes with T and G always goes with C. Because each strand can be used to make another strand, the strands are said to be complementary. Now DNA is replicated or copied during a stage of this thing called the cell cycle. And we're going to talk about the cell cycle in detail next week. The cell cycle is how cells actually make new cells. And so DNA has to be copied because each body cell gets a complete set of identical DNA. And this is important because that DNA holds all the instructions for making you. And different cells need different parts of these instructions in order to do their jobs. So DNA is replicated during the S, or the synthesis stage of the cell cycle. And again, we'll talk about that more in detail next week. We have 3 stages to DNA replication. Now, those of you that are working online at home, please make sure that you've downloaded the little handout that goes with this PowerPoint, because I have these 3 pictures here and I have this set up in stages so you can copy stage 1, stage 2, and stage 3 onto that handout so you've got pictures to go along with. So in stage 1, we have our 2 strands of the double helix unzipping, just like a zipper, okay? When our zipper is connected, it goes all the way up. We have the 2 sides to it. When we unzip it, we have the 2 sides that are separate. And when they separate, we have these things called the replication forks, okay? So our DNA unzips, forming replication forks. Now, in step 2, once we have those replication forks, new bases are added following the rules of base pairing. A is with T's and G is with C's. And we have a special enzyme that comes in and joins those nucleotides. That enzyme is called DNA polymerase. So this is the one that brings the A nucleotide or the T or the G or the C. Then we've got step 3. And in step 3, basically, we've completed our stages of replication. So we have 2 new molecules of DNA being formed, each with an original strand or parent strand and a newly formed strand. And because it has an original strand and a newly formed strand, we call this semi-conservative because half of the original DNA molecule is still there. Now, replication is very fast and very accurate. And in eukaryotic cells, because there's so much DNA that needs to be copied in eukaryotic cells, it starts in multiple spots on the chromosomes. Okay, chromosomes are just tightly compacted pieces of DNA. And so if you look at my picture there, I have A, B, C, and D. In A, we have our strands of DNA. Okay, the little circles there in the middle are showing where replication is going to occur. In B, we have it forming the replication forks. New bases are getting added in C. And then finally in D, we have our 2 new molecules of DNA. Remember, it's semi-conservative, so each new molecule has 1 original strand and 1 new strand. And DNA polymerase also can find and correct errors. So if it's bringing a nucleotide, it kind of gets double checked before it says it's ready to go. In prokaryotes have very, very little DNA. They only have 1 circular loop of DNA. So replication starts from 1 single point and proceeds into 2 directions until the entire chromosome is copied. Now, one interesting thing to note is that During replication, DNA can get lost from the very tips of the chromosomes, okay? Remember, chromosomes are just tightly compacted pieces of DNA. And these little tips are called telomeres. And this loss of DNA from the tips is involved in the aging process. So every time your cells get copied and DNA gets copied, a little bit gets lost. And these chromosomes get shorter and shorter and shorter as you age. All right, and that is it for our presentation on DNA replication. We're going to talk next a little bit about RNA and some other processes in how that gets turned into proteins. If you have any questions, please let me know.